Our next speaker is Imre Holb. Imre is a professor of plant pathology in the Center for Agricultural Sciences at the University of Debrecen in Debrecen, Hungary, and the Hungarian Academy of Sciences in the Institute of Plant Protection. His professional responsibility is education, teaching, research, and extension and outreach in plant protection for fruit crops with an environmentally friendly approach, both integrated fruit production as well as organic fruit production systems. Currently, he is the associate editor of the European Journal of Plant Pathology. So this time, it's our pleasure to introduce Imre.
So thank you very much for the invitation. And well, our research group working on environmental friendly fruit production and especially on plant protection and including epidemiology forecasting and disease management. In this lecture, I try to focus on organic fruit production and try to show you how we explore new epidemiological aspects, forecasting, and also how we incorporate into disease control strategies. So first, see the tendencies and the problems in fruit plant protection. A few years ago, more than 10 years ago, from the United States, it was published a paper in the Journal of Nature about the sustainability of three apple production system, and it has three main conclusions about the plant protection. The first one was that the environmental friendly benign production system, and especially organic, received great attention by the consumers, and especially organic production need new disease as pest control methods, and also inserting new knowledge of population biology features in management practices. And the third one, the problem is there is a great difference in disease and pest management efficacy of the three management systems. What is these differences? First, in integrated, some synthetic products are restricted. In organic or synthetic products are banned, and only natural products are permitted. And as a consequence, disease control against fungal diseases is less effective, especially in organic orchard, than in conventional production. So the basic requirements is quite easy. What we have to do to improve disease management efficacy in organic orchards. So my first question was more 15 years ago, what crop we used. In the last night presentation, we could see berries, apple, and bananas. In Hungary, we don't grow bananas yet. And berries is not a big production, as Franco told us, so remained apple. And if I use apple, then I should use the main fungal diseases, which is a key organism and also can be used in model organisms. So this was apple scab. Unfortunately, here has no meaning of apple scab because you don't have apple scab. But sorry about that. We have a lot of problems with that, so I will talk about this disease. But you will see the programs, what I provide here, will be used or can be used, at least the strategy for other diseases. So the first aim was to investigate the epidemiological aspects of inoculum sources in environmentally benign fruit production, especially in this lecture in organic. And the second one, we try to transfer this knowledge into the disease management practices. So what we have done, we started background information, disease cycle in spring for apple scab. But we know for a long time, the disease is overwintering on the fallen infected leaves. And next spring, it produces a sexual form of perithecia or pseudothecia, depending on the literature, and forms ascospores, and ascospores infect the leaves. So this is what we know from the past. And what we did, we tried to reveal all the things in organic orchard, and what we realized first, the area under disease progress curves was 10 to 50 times higher in organic orchard compared to the integrated ones. If you look at the figures, is the x-axis is the time, and the y-axis is the disease incidence, and you see organic is always much higher than the integrated. So, and area under disease progress curves is representing this. So scab symptoms, and that was the key point, in organic orchard appear or observed earlier, already in green tip stage than in the integrated orchard. So you kids could see in organic orchard the symptoms was much earlier than in the integrated orchards. And normally the ascospore discharge was nearly the same date and time in both systems. and it's occurred at warmer winters. So it mild winter strategies. This question was that why is this phenomenon and what is the cause of this? So we had at that time two hypotheses for that. The first one, the pathogen comes from the neighboring orchards. 30 years ago, it was an idea about that. Ascospores has 5 kilometer spreading distance from the outside orchard. So what we have done, we selected an orchard which was more than 5 kilometers from others. So we exclude the inoculum from this orchard. But the second one still remained, how we can eliminate ascospores from the overwintered leaves effectively within the orchards. So what we have done, we made a sanitation practice. We collect leaves and then we cover the soil with plastic foil. And so we can eliminate all these things. So the only things what remained, probably the pathogen overwinter on the tree. And therefore we made an experiment, a sanitation experiment for both integrated and organic orchard to see how does it works. So let's see first the integrated orchard. In integrated orchard where the scab lesions on 100 leaves or shoots in autumn was only 10, so it's a really well treated orchards. And if you look at the discharge in time, starting in green tip stage and ending at the beginning of June, and if we look at spores, ascospores, this is the red points with the spore trap data, you can get spores there. And after a 2 weeks incubation period, you get scab lesion. If we removed the infected leaves from the orchard floor, then we don't get ascospores at green tip stage. And it is not surprising, we don't get lesion also. So what happened in an organic orchard? You see 500 times more scab lesion on 100 shoots in autumn. So if we don't do the sanitation, we have the ascospores. And of course, we have the symptoms, much more symptoms than in the integrated orchard. And when we have done the removal, if we do the removal, we do not expect any symptoms, yes, because we remove the leaves and the infected part. So we don't have ascospores, but surprisingly, much earlier we have a scab lesions. So the question arose, from which part of the tree does the infection come and what inoculum sources can be on the tree? From all the literature, the crackings of woody tissues can be infected by conidia, but we could not find this kind of sources. And the other one, also from all the literature, is mycelium and conidia can be on the shoot surface. We find only very few, and then it was not significant. And finally, what we find much more is conidia inside the bud scales. And when you look at it, you get more data on that case. And these inside bud scales germinate inside in early spring and then cause early symptoms already in green tip stage. So we find where is the inoculum source on the tree, and it's inside the bud scales, but we still have some questions arose. One of the questions was when and how do conidia get into the bud scales, and the other one, how many overwintered conidia needed to cause significant symptoms in the next spring. So let's see the first question. So we have the bud scales in February, but probably it is open in the previous season. So when it's open, the conidia can get inside. That was our hypothesis. So let's see what happened. So what we did, we collect every week bud samples, and we look at inside the bud scales if there is conidia or not. And you see, at the end of the season, increased the number of conidia inside the bud, and at a certain point, it was just leveling off. So the cumulative number of conidia did not increase anymore, and this level was the bud closure stage. So at a certain point, the bud closed, and if it's closed, no conidia get into inside anymore. And one of the things what we also realized after rain events, and it was tested in laboratory, then inside bud scales. So when the rain comes, then it's washed inside the bud scales. So it helped to be infected the bud scales. So we get the answer how the conidia get into the basket. So it's a previous season infection, or not an infection, but at least a washing inside the basket. So the second one, how many overwintered conidia needed to cause significant symptoms? So we have in autumn this buds inside the conidia, but it has to overwinter, and we have to see how much infection can make the next spring. So what we did, we measured the autumn scab incidence, and it was mid-October, and to see next spring how many conidia inside. And we measured in the bud outer, inner, or the bud inner surface, and also shoot, and the viability of this conidia. What you could see exactly, you see it's 40%, and before 40%, really few amount of conidia, but after that it's a really large amount. We did several statistical analysis, and we set up at this threshold level, and this threshold level was the autumn scab 40%. So if you have an orchard above 40% disease incidence at autumn, sure, next year you can have an infection by overwintered conidia. And this samples was made more than 20 orchards all over Europe, and 90% of the orchard was organic. And that's the big meaning of that. So most of the organic orchard is infected by scab. And the reason is most of the organic orchard is use moderate or scab susceptible cultivars, as Franco told in the previous lecture too. So get further on. We have several knowledge. So how we can control overwintered conidia? That was our question, several years ago. And one of the hypotheses was that copper fungicide are the most commonly used fungicide at dormant bud stage and bud burst stages. And when the bud bursts, then open the bud scares, it's spring. So copper can go inside and can kill this conidia, which overwintered. That was the hypothesis. And the other one is what we realized under our study, 80% of the conidia were found the upper third of the shoots. So if we cut this part of the shoots, probably we remove this conidia. So let's see what happened in the copper spray treatment. So what we have done, you see here an orchard part. This black part is the source of inoculum. This white part is the covering with foils. And it was a 60-meter part. It's covering the orchard with foils. That was 1 hectare orchard, an experimental orchard. So what you see in the figure, this is the 0 point. This is the black part. And these distances is the white plastic foil covered part. So we did 2 experiments. One was the untreated, and the other one is treated at bud burst with copper. In the untreated one, that you get is infection from the ascospores and the conidia, as you don't use the copper, and with distances, it's decreased the amount of symptoms. If we use the other one, the copper's one, we kill the conidia, so we get another curves with ascospores. So between the 2 curves, we have the conidial infection. And if it's true, then the copper spray can kill a sort of conidial infection. And further studies prove that the recommendation for using the copper spray when the autumn scab incidence was between 40 and 60%. Okay, let's see about the winter pruning treatment. In the winter pruning treatment, what we have done, we cut the upper third of the shoot, but we used 2 types of pruning. It's a weakly pruning and a strongly pruning. We use susceptible varieties, a moderately susceptible, and also a VF-resistant cultivar. And what you could see, strong pruning always have significantly less incidence on scab in April and the other one. And further investigating this, that we realized that above 60% combining with the copper spray, it can be used as a tactic, as a control strategy against this overwintered conidia. So we have several information and control strategies, but we had problems as copper was banned in several Western European countries. At that time, I worked quite a lot in the Netherlands, and in the Netherlands already copper is banned, and in most Scandinavian countries. So the problem was that no effective fungicide control in organic production against Scab at that time, and copper fungicide method against overwintered conidia getting be in a trouble. So we need new alternatives. At that time, the available things that we had is we want to be sure if we find a product which similar to copper in phytotoxicity and also in efficacy, and to see how does it works this against this overwintered conidia. So what we have done, we tested several products available at that time: vegetable sulfur, copper hydroxide, vegetable sulfur, and calcium polysulfate, lime sulfur. And what you could see, the lime sulfur component was more or less the same in efficacy and phytotoxicity than copper hydroxide. And we find it as an option for copper replacement in that case. And what we have done also, in one case, is we were late for spraying. So we used calcium polysulfate as a post-infection spray or after infection spray, and surprisingly it worked quite well. So we did another test, and in this other test we realized that the preventive copper spray was as good as than the after infection sprays with calcium polysulfate 15 and 25 hours after infection. And if you see, it was in 2003, and this was the first time when you could use a forecasting system for organic production. If you see the plant protection products for organic, you could see that most of them is a contact product, so you can use them preventively. Later on, as Frank also mentioned, the potassium carbonate and other products has also after infection activity, and could be used as we find out for lime sulfur in that time. So this is what we gained: calcium polysulfate and with post-infection activity, previous autumn scab threshold, winter pruning, and bud closure stage. So what we could do with this? Okay, integration into organic scab management strategies. And what was our strategies? First, we tried to make disease progress curves and derive the most important parameters. And these parameters were first, the Y75, which represent the blast causing stage, and then the absolute growth rate and the area under disease progress curves. So we try to insert all these parameters into computer-based forecasting systems, like the pathogen submodels of these systems. So we include this Y75 stage and autumn's threshold, and also the minimum values of the area under disease progress curves and the absolute growth rate until the trees reach the day 75. And if you collect this data, you can fit the model, and the logistic model was the best. And after that, when you fit the model, the computer calculate for you the values of our DPC and this absolute growth rate. And if it is lower than this minimum that we give to the model, and the autumn scab incidence is lower than 40%, then you don't need spray against overwintered conidia. If this value, the calculated value, higher than the minimum, and the incidence between 50 and 40%, then you have to have a fungicide spray. It can be copper, lime sulfur, or potassium carbonate, or whatever, with a kind of activity. And computer-based calculate if the minimum, and after that it's really high, then you should use a fungicide sprays with winter pruning. But all these things about overwintered conidia, we have still the problems of ascosporic inoculum, and we want to also include this. So we have to find the best methods which remove the fallen leaves, which contains many things in it. So if you look at this picture, what we done, we use lime sulfur spray on the soil, mulching, or we collect leaves. We combine these techniques together, and the best one was the leaf collection treatment, or the leaf collection treatment with all the other ones, mulching and covering the orchard floor. We also tested biological control agent, but was much worse than all the other leaf collecting treatments. So we were not implementing in our tactics. So some new question arose. Can the spray number be reduced by using this sanitation practice and the overwintering conidia methods, control methods? And the second one, can the spray number reduced at the end of the season? And the third one, how scab susceptibility of the cultivars affect all these things? What we set up first, we used inoculum removal, no inoculum removal of leaves with leaf collection and pruning. And we started 3 stages: dormant bud stage, early tide cluster, and pink bud stage. This is the starting date of the spraying. And the finishing date was mid-July, mid-August, and mid-September. And used low susceptible and moderate scab susceptible cultivars. And what you could see here is that for the moderate scab susceptible cultivars, for mid-August, if you use leaf collection methods with pruning, then you could see quite a low amount of disease. But if you use the low scab susceptible cultivars, then you can stop spraying already in mid-July, and you don't need to use any inoculum removal. If I put it in a time schedule, you could see that in a moderately scab susceptible cultivar, you can start to spray an early tide cluster, and you should finish it at mid-August, but you should use leaf removal and prunings. And for the low scab susceptible cultivars, you should use it a pink bud stage starting date, and the ending date is mid-July. You should use the forecasting against overwintered conidia and without removal of inoculum source. So we made this complex control strategy against scab for organic orchards. And what we have done, we compare a general spray schedule and the complex control strategy, and to see that how does it really works in the practice. Of course, we did it for integrated orchard. There's a different story, but here I show you what was the result on this one. And the new strategy decreased the number of spray. And for organic, what happened is the general spray against scab is 25. And if you use the complex control strategy, it was 20. And as you see, the number of spray can be reduced 5 to 25%. So one of the remarks here, most of our orchard contains moderate or susceptible scab cultivars. This is why so high the number of sprays. But to reduce the number of sprays, that was the big issue in that case. So this is the story for scab. But we have other problems. With the Monillia fructigena, I will not tell this story, but it was similar. And the basics of all things are similar. So we have made the epidemiological part. We made PC, program-based things. We made threshold. We made the intensity level. We used the fruit injury aspects. We used the drop fruit aspects. And we also provided a strategy for similar than as you see in scab. And laterly, so this is for organic orchard. Of course, we did it for integrated one, but for integrated, Monillia fructigena is not really a problem. So what we have also done, we selected all other diseases, the 5 most important diseases, and we made an overall disease management strategies. This is more complex. And this was published by myself in the Sustainable Agriculture Reviews. So you can find it in a 30 pages, things how we could do things together and what was the concepts to moving from integrated management to a certain organically managed production system. So this is the end, and I would like to acknowledge to my colleagues in Hungary and the Netherlands and from the US also and in the UK and the several programs which helped us to develop this kind of things against mainly scab, but also about other diseases. So thank you for your attention.